So first we have Archishman Biswas from here KTH talking about dynamical consequences of non-random connectivity neural networks. Go ahead.
So, yeah, welcome back everybody. So yeah, I'll be talking about my current PhD project that I have been doing, and it's on this topic, dynamical consequences of non-random connectivity in neural networks. And yeah, the title is very generic, so I will start going into the details gradually. So the first thing I would like to talk about is the scale at which I am trying to look at this phenomena, because yeah, many people in this workshop have been showing modeling across different scales. But for me, it's if you zoom down to the cortex and then you reach a cortical column, then you see this cortical column is made out of excitatory and inhibitory cells. So this is, for me, when I say a network, this is the network which is made out of excitatory and inhibitory cells. And so each node would be that one neuron, and the edges here are the synaptic connections between these neurons. And the main observation that has there been in the past is that the brain connectivity or the connectivity in these cortical neurons are non-random. And many people have used different network modeling strategy to capture that non-randomness in this brain connectivity. So yeah, different connectivity models have been used to capture the structure of the microcircuits. And here I have just given some examples to begin with. For example, people use these Erdos-Renyi networks or the random networks, which basically captures only the average connection probability. Then there are things as distance-dependent connectivity, where people also take into account the spatial location of the neurons, because these neurons are located in a 3D space. And then you also have things like clustered connectivity, where connectivity depends on, like, the connectivity between two neurons depends on the type of the two neurons. So there are connections in the class and less connections outside the class. But the main thing is, like, across all these three matrices, the average probability of connection is same. But still you can see that the overall structure or how they are arranged changes a lot. So yeah, this is one way of one way of appreciating that there, even though the overall connection is same, overall number of connection is same, but still you can have certain non-random patterns like seen in these two. But what I will focus on other related to some other observation, which is related to capturing non-randomness using motif counts. And the overall story starts back in 2005 when this paper came out by Song and other authors. In that paper, what they did is they did this quadruple patch whole cell recordings from rat visual cortex. So they simultaneously attached these recording probes to 4 neurons, and they stimulated one of the neurons and saw the effects of that stimulation on the other neurons. And they were doing it in pyramidal cells. So TTPC is thick tufted pyramidal cells. So these are big cells, and it's easy to put your like the recording probes on them. And yeah, if you do it, then what you can basically get out is the synaptic connections between these different neurons. So for example, the synaptic strength that goes from neuron 1 to neuron 3 is 0.5 millivolt. The one that goes from 3 to 1 is 0.3 millivolt. And so the exact strength is okay, but the main thing that I will focus is rather the presence or absence of these edges. For example, in certain cases from 1 to 2, you might not even have this edge. So now the next aim is to capture the non-randomness, and I will just show one example of how it's done for one particular motif. And then I will show how it extends to other things. So for example, if I want to see what's the probability of this reciprocal motif, which is a connection that goes from A to B, and simultaneously a connection existing from B to A, and compare it to the random case, the way to do it is as follows, that you measure the probability of one edge existing independently of anything else. So there you can get a probability number, like for example 10%. So you get a P, and if you assume a random network baseline, what you will think is that the probability of this reciprocal motif existing would be P square. But then from these observation, you also make the actual, you can also find the actual probability of such reciprocal motive existing. And then you can compare these two numbers to find out what is the prevalence of the reciprocal motive, or how more prevalent or less prevalent is a reciprocal motive compared to the random network baseline. So this I did for one particular motive. You can do it for all other kind of triplet motifs which are shown here in the x-axis. And yeah, you can plot that ratio of the actual versus the expected from a random network baseline. And yeah, you can get this curve. And in the random network case, everything would be 1 because yeah, that's the expected value. But when you see the deviations, then you basically characterize, it becomes one way to characterize the non-randomness in the networks or the cortical networks that we see, the non-randomness there. So that is the one of the earliest study, but yeah, we have more recent studies too. For example, this one was done in 2020, published in 2020, where now the thing that has come up is that you can do you can put more probes. So there are 12 parallel recordings. So it is 12 patch whole cell recording from L5 thick tufted pyramidal cells in the rat somatosensory cortex. And there they also show those kind of non-randomness that is characterized by the motif counts. And they also give a review of past work which talks about the universality of motifs, that across different studies or different methods or different brain regions or different cortical regions, you see the overrepresentation or underrepresentation of these motifs. And some are like, yeah, there will be some motifs that are similar to what is expected with a random network baseline. So yeah, now these things varies a lot across studies. So yeah, I will not talk about the exact count, but the main thing to notice is that yeah, there is non-randomness that can be characterized by these motif counts. And so these are rather electrical recordings, but even if you go to microscope recordings or microscopy-based recordings, where you look at the image and then you see the connections. So regarding that, a recent work that was published last year, which is regarding the flywire connectome, that also shows the non-random connectivity. And the similar graphs that I was showing, similar things to that you can see in these red crosses here, which again compares these motif counts to a random network baseline, which is here in the dotted line. So yeah, you still see the non-randomness that is captured by these motif counts here. So yeah, this is mainly regarding the observation that we see what kind of connectivity patterns we see in the cortex or even different brain regions of different animals, like even in simpler brains. So that's one thing, like the observation of motifs. So now I would start with trying to understand that how these local motifs rather translate to global connectivity, because these local motifs are things that we are measuring locally, but we don't, just by looking at them, we don't completely understand how the overall connectivity matrix looks because of the presence of these kind of motifs. So here I will only look into these 4 particular motifs, 4 triplet motifs, which are this reciprocal, convergent, divergent, and chain, and try to see how the matrix turns out to be if I try to construct a connectivity where these motifs are overrepresented than compared to the random network baseline. So if we do that, we get these kind of connectivity matrices. So yeah, in the reciprocal you can't see much, but because the matrices are quite sparse, it's only 10% connectivity. But the main thing that happens is the matrix gets more and more symmetric. In the convergent case, you start seeing these kind of horizontal stripes, which means that the in-degree distribution is more variable compared to the random case where it has a binomial distribution. But here the variance will be higher than the binomial distribution. Similarly, there is the divergent case where you see these vertical stripes, and here the out degree variance will be higher than the binomial case or the random network case. And finally, you have the chain motifs, which when you induce into the network, then what it does is basically correlates the in degree and the out degree distributions. So yeah, so finally the convergent motifs lead to higher in degree variance, and divergent motifs lead to higher out degree variance. And finally, the chain motifs are related to the correlation between the in degree and the out degree distributions. Now, yeah, this will be explained better in this if we just look at it visually. So here I'm showing certain plots where each dot here represents one neuron and or one node in general terms. And for that one node, I can calculate what's the in degree of that node and what's the out degree of that node. And then if I make these scatter plots and also see the distributions of the in and the out degree separately, I get this kind of plots. And this is for the random network case, where each of these distributions will be binomial and for the in degrees and the out degrees. Now, again, we can look into the specific case, for example, the case of convergent motifs, which is here, and the corresponding matrix look like this, connectivity matrix looks like this. There, if we see, then there, if we make the same plot, we see this kind of patterns, where now the in degree is much more variable compared to the binomial distribution case. So yeah, what I'm basically trying to show is how these local rules translates to the global connectivity case. And so the main statement is that convergent and divergent motifs result in variance that is higher than the binomial case. So this is for the convergent case. If you do the divergent, it just flips. So you can imagine that it's just a transpose of both these diagrams. Now, maybe I should move on to the chain motifs, where when you add chain motifs, what you are basically doing is either, you are putting a correlation between these in degrees and the out degrees. So on the left side, I am showing the case for negative chain. So where I have put negative chain motifs. So there the chain motifs are less compared to the random network baseline. So there what happens is that the in degrees and the out degrees, these two distributions are now anti-correlated. Whereas in the case of positive chain, the in degree and the out degree distributions are positively correlated. So the marginal distributions remain same for the in degrees and the out degrees. The only thing that changes is the correlation between these two distributions. So yeah, again, the positive chains lead to correlated in and out degree distributions, and negative chains lead to anti-correlated in and out degree distributions. So yeah, these are one part of my study that I have been doing. And in general, I'm also trying to study these things, that how local connectivity leads to, how local connectivity rules leads to certain global connectivity matrices. But the next thing, or the main part that I want to discuss, is rather regarding this, that how motifs affect the dynamics of cortical circuits. So yeah, there are many ways to model the cortical circuits, but the way I rather use this using this excitatory and inhibitory neurons, where where I think of the cortex is made up of this excitatory and inhibitory neurons, and then these neurons form a network, and in those networks now I put in these connectivity matrices which has those motifs inherently in them, and then I try to see what kind of dynamics come out, and then I try to compare that dynamic with respect to the random network case. So this is basically the model. I have put in some details, but that doesn't matter much. The main thing is that it's a population of excitatory neurons, which I'm calling the pyramidal cells, and there are inhibitory neurons, which are also called, like, the most prominent category is the parvalbumin cells. So PC is excitatory, PV is inhibitory cells. So hence we have this excitatory and inhibitory cells network, and there are this inside there are connections between the excitatory cells and the inhibitory cells, and there are also these interpopulation connections which are kept random. And then what I do is I gradually change, or I one by one I change the matrix that is between the that is inside the excitatory subpopulation, and I see how the dynamics change. And the dynamics here I'm showing in terms of raster plots. So this is spiking activity across time for the excitatory population in red and the inhibitory population in blue. So this is the case for a random network case. Now, what happens when we move to convergent motifs, or when we induce convergent motifs in this excitatory subpopulation, then we start seeing these kind of patterns where you have quite high synchrony compared to the random network case. So here we can see these kind of synchronous events that arises. And then finally, if we rather look into the chain motifs case, there we see that the synchrony synchrony increases much more compared to the random network baseline. So there we can get an idea that these kind of motifs, when they are induced into this circuit's connectivity structure, it creates certain, it changes the dynamics. And here I'm particularly looking into synchrony. So the main thing that I find out is that higher convergent and chain motifs in the E population leads to higher synchrony. And here I was showing the qualitative things, but yeah, you can also like use some easy measures for this for measuring this synchrony. And one of those measures is Fano factor, which is basically the variance upon expected value of the aggregated rate of this activity. So if I now plot this Fano factor across different cases of these motifs induced in the excitatory population, I get the following curves. So as I showed earlier, that there are those 4 motif structures: convergent, reciprocal, divergent, and chain. Hence, if I do 2D plots regarding each of these motifs, I will get these 6 plots. And the color here shows the Fano factor or the synchrony. So the higher the Fano factor, higher is the synchrony. Now one can go through each of these figures, but the main figure is related to this, the chain and the convergent motif, because what I observed is that it's the chain and the convergent motifs that has a higher impact on the synchrony of the network. So yeah, if we just look at this figure over here, which is the chain versus convergent motifs, and then how the Fano factor changes, we can see this, that higher convergent and chain motifs in the E population leads to higher synchrony. So basically, if you move up this diagonal like this, like if you go to the top right, then your synchrony keeps on increasing. So again, chain motifs and convergent motifs have a synchronizing effect. So now the main thing that I want to come to is that these motifs induce synchrony, it's all right, but this kind of synchronous behavior in the spontaneous activity of like cortex it's generally not observed. So this is something, this synchrony is something that is not observed in the cortical network dynamics. So yeah, the main question that then we can ask is like, what are we missing? And the main thing there is that it's the motives in the inhibitory population. So till now we have added the motives in the excitatory population, but now we also need to look into the effects of the motives in the inhibitory population and what effects they have. So that is the next thing I have done, like adding motives in the inhibitory population. So for that, I also need to put in these matrices or these motifs in the inhibitory population. And there again, I'm starting with the raster plots for the high synchrony case, or when there were chain motifs in the excitatory population. So there you get this synchronous kind of event. Now, if I put some convergent motifs in the inhibitory population, I get the following raster plots. So now the synchrony has been reduced down. It's still not very low, but there are still some synchronous events happening like here, but still it reduces down compared to the earlier case. Now, if I put in chain motifs here in the inhibitory population, it reduces down even more. So the overall firing rate is also reducing, but still the whole activity is becoming less synchronous. So that is the main takeaway here, that motifs in the I population quenches the synchrony. And this was again a case-by-case qualitative thing where I was showing the raster plots. You can also look at the Fano factors and compare them in a graph. And that is what I have done here. On the x-axis, I have the cases for the motifs in the pyramidal cells or the excitatory network. And in the colors here, I have the individual cases for the motifs in the parvalbumin cells or motifs in the inhibitory population. And there you see like for each of these cases in the pyramidal cells, the Fano factor reduces as you start inducing motifs in the inhibitory population. So yeah, that's the main thing, that it has a reverse effect. And like the diagram I was showing earlier for the chain versus convergent of the excitatory population, you can do the same thing for the inhibitory population. And there you can also get the similar diagram. But now what happens is that in this diagram for the I population, it's reversed. Like now if you go up this diagonal, the Fano factor keeps reducing down. Yeah, it keeps coming down if you go towards the top right along that diagonal. So yeah, so these motifs have an impact, but in the excitatory and the inhibitory parts, the impact kind of reverses. So that's one thing. And finally, I would like to show one result, which is it's a result for a particular case. It's not a general thing, but in one of the simulation runs, what I have done is try to look into the competition between suppose the chain motifs in the excitatory network, which is here in the y-axis, and the convergent motifs in the inhibitory network. So each dot is one particular simulation run for one particular combination of the motifs. And then, I have fitted some curve to get the whole image. And then you can see these certain contour lines. And particularly if you look above this red line where these chain motifs start having a synchronizing effect. Here, if you start inducing the PV network convergent motifs, the synchrony reduces down. So they have a competing effect. So if you go right, the synchrony reduces. And yeah, you can do the same things. You can get a similar diagram for this PC chain motifs and the PV chain motifs. And there you can also see the competing nature of these motifs based on which population you are inducing these motifs in. So there also you can see these contour lines, and maybe along this direction, if you have motifs along this direction, your synchrony won't change. Although your networks are changing, the synchrony won't change. And synchrony is just one measure for the dynamics, but still, like we can say in some sense that the dynamics is not changing as we move along this line. So that was all the parts. I will now just share some key insights, and I have tried to keep it like a bit general. So the first thing is that only certain network features have significant impact on the dynamics. Here I was only looking into 4 motifs, but even there, the reciprocal and the divergent motifs didn't matter much, or the variance in the out degree or the symmetry in the network, it doesn't matter much. It was rather the convergent and the chain motifs that had an impact. And the way I was generating these networks, I later realized that having these two things together also leads to this generation of the feed-forward loop, because if you can see, this is a chain like through the I, and there's also a convergent case in that case. So it can be thought of as a combination of all the convergent, the divergent, and the chain motifs. When you have all those things together, you get these kind of feed-forward loop. And other people have shown in their talks that it's an important thing to measure. And for me, maybe this motif and also higher order simplices, they might lead to higher synchrony in the network. So that's one thing, that only certain motifs matter. The next thing, that the dynamical changes will depend on which population the motifs are expressed in. So either the motifs or some other network features. So it depends on which population it is in. So is it in the E population or the I population? And even in inhibitory population, you can have different categories of inhibitory populations. So it will depend like on in which of the populations you have these motifs. That will decide the dynamics. We can't just talk about motifs, but we also look into the cells, like in which neurons it is being observed. And finally, it's a general statement that the measurable things that we have are rather regarding the local properties or local connectivity rules. And we should think that these local properties rather affect the global connectivity of the global connectivity in the whole network, which is captured using these connectivity matrices. And that is something that rather is important for understanding the impact on dynamics. So yeah, we should rather think about this arrow, how these local rules affect the global connectivity, and also maybe think about a reverse arrow that a reverse arrow that goes from global connectivity to local rules, and which might help experimentalists to search for these kind of local rules. So yeah, that's all about my presentation. So yeah, if you have any questions, please ask.